We hit the 6 o'clock, so I'm going to go ahead and get started. Let me introduce myself. My name is Erin Deely. I'm the Director of Recruitment and Engagement at the College of Science and a proud alum. If you were here earlier, you already heard me talking about that. And I'd like to welcome those of you that are returning from the two previous live virtual Science Cafe talks that we've had in the previous weeks. If this is your first time, welcome as well. Many of you may know that we typically host, or we have had a longstanding series of Science Cafes at community locations. There are community partners. And so our speaker tonight was featured at the Saddlebrook Science Cafe series that was, going to be, that was scheduled for this spring. And previous talks were from other venues. I also just want to, we wanted to adapt to the inability to have these talks in person. So these three speakers had agreed to provide this as a live virtual Science Cafe format. So we're really grateful for that. So I just will mention again that it would be best if everybody would use the question and answer function at the bottom of your screen. If you hover over the bottom, you'll see that Q&A icon and that you can submit your questions there. We'll address questions after the presentation, just for flow and ease. And I'll do my level best to have questions that were asked first. So first come, first serve on the questions. Obviously, if there's duplicate questions, we'll pass over that. And I want to give everyone that submits a question the opportunity to ask. So if you have multiple questions, I may not get to all of your questions initially. Just so you're aware. And then again, the format of the talk is the presentation will go for about 40 minutes, 45 minutes, and then we'll have question and answer. And then following this talk, it'll be a few days, probably early next week, we will upload the recorded presentation to the College of Science YouTube channel. And so if you visit, I posted in the chat, you can look at the chat panel. I posted our website, the Virtual Science Cafe series webpage. If you go to that link, next week, then you can click on, you'll have a link that takes you directly to those YouTube presentations. So just be mindful that we'll have those there. And then the Science Cafe series at our community partners or public locations are postponed indefinitely. The College of Science is going to be rethinking how we do this, at least for the fall. So stay tuned on that website. We'll update folks about what we're doing. And if you're on our listserv and receive email announcements about these talks or follow our social media accounts, we'll update via those venues as well. Well, without further ado, the reason that you're here, I want to introduce our speaker. Dr. Sutphin began his academic career as an engineer, earning a BS and an MS in aeronautics and astronautics from the University of Washington in 2004 and 2006, respectively. He received his PhD in Molecular and Cellular Biology from the University of Washington in 2012. And completed his postdoctoral training at the Jackson Laboratory in 2017. He joined the U of A as an appointed assistant professor in molecular and cellular biology in 2018. And his research focuses on understanding the molecular basis of aging, hence the title of his talk, and by understanding the basic molecular architecture that underlies the aging process, the goal is to identify new targets for clinical intervention to extend healthy human lifespan and simultaneously treat or delay onset of multiple disease of aging, such as neurodegeneration, cancer, cardiovascular disease, and diabetes. And I asked Dr. Sutphin to just provide a couple of, personal fun facts, and what he shared with me that I'll share with you is he practices what he studies. He's tried several forms of dietary restrictions, one method to extend lifespan, and currently practices intermittent fasting. And his dog, Doppler, is contributing to aging research. Yours can too. And it is my great pleasure to welcome Dr. George Sutphin for his talk, the science of getting older. My apologies. Without further ado, go ahead.
All right. Thank you, Aaron. And thank you all for being here today. I hope you're all staying relatively healthy and sane in the current situation. I'm glad we were able to make this happen, even though we couldn't all be there in person, which I'm sure is all of our preference. As Aaron mentioned, I was previously an aeronautical engineer. That was what my initial training was. But I ended up switching to biology because I developed kind of a passion and a fascination with the problem of aging, something that affects all of us. And particularly, as we'll talk about in a moment, this is kind of an opportune moment to be studying aging in our society and in our current point of advancement. And so I'd just like to kind of share with you today some of what we've learned about aging and how, as scientists, we think about the aging process and some of the things we're trying to do to mitigate it and what we hope the benefits of that will be. Just to give you an orientation to the talk, I would like to first start with a fairly brief look at how we as humans have thought about aging and longevity historically. Go into why right now is a good time to study aging. Talk about what aging really is from a biological perspective, and then maybe give some discussion of what we can do now, what we know now we can do to age successfully, hopefully remain healthy into our later years, and maybe extend lifespan. We'll see. We don't have all the answers to this yet for sure, but maybe talk about what the current state of the art is in some of that area. So what have we thought about aging historically? If you look back through human thought, human literature, we've always been fascinated with this topic. This is certainly not a comprehensive overview, but I just wanted to highlight a few kind of the myths and legends that are out there that highlight long lifespan or aging, or immortality is the common kind of myth for this sort of thing. The very earliest literature we know about, in the Epic of Gilgamesh, which dates back to 2600 BCE, this was kind of in this first earliest literature, we have stories of the quest for immortality and longer lifespan. There was a character, Unapishtim. I tried to practice that, but that one's a little more complicated. So he was one of the survivors of the Great Flood. This is another one of the many flood myths in our early literature from the Middle East. And he was granted immortality, and Gilgamesh sought him out to learn the secret, and went through a series of trials, which he ultimately failed, of course, for those of you who know the story. So even from our very earliest literature, they were doing this. In Greek mythology, there's various stories of gods granting eternal life. Prince of Troy, for example, who was granted eternal life, but not eternal youth, which is a point of worry that is often brought up when I talk about this topic. What if we can live forever, but we don't solve all the health problems? That was an early theme in quests for immortality and longevity. The biblical tradition is chock full of very long-lived individuals. If you work out the dates, from 123 years, which is older than our current oldest verified record, which is around 122 years old, up to Methuselah, who is the most famous for this, at almost 1,000 years old. Nicholas Flamel, who is an alchemist, purported alchemist in the 1300s to 1400s in France, was purported to have discovered the kind of ultimate quest for alchemy, which is the philosopher's stone. This is the substance that's supposed to turn lead into gold, and also give you eternal life, have properties that give you the elixir of life. So even from our very earliest literature up to kind of modern themes, we see this kind of theme going through of people looking for ways to extend kind of healthy lifespan and improve the number of hours we have on this planet. If we look a little more recently, and here we're going to jump way forward up into the early 20th century, which is where we first started to really develop some methods that actually look like they might work to do something like gain a longer, healthier lifespan. Aaron already mentioned this concept of dietary restriction. So what we mean by that is reduced food intake in some form, but without malnutrition. So you're still getting adequate nutrients to kind of maintain health. But you're taking in either fewer calories or fewer specific components of your diet. So this kind of idea of general dietary restriction was first observed in the early 1900s. There were a couple of papers that found if you take a rat and reduce how much food they're allowed to eat, you can get a reduction in how often and how many cancers the rats get. So this is the first idea. Just a little bit later, this Clive McKay, that was the scientist who discovered this at Cornell, not just looking at cancer, but actually at how long the rats live. We're going to talk a lot about lifespan extension in this talk, and the reason for that, aging is complex and it affects virtually every system in our body. All of our organ systems, all of our cell types, many of the kind of underlying molecular processes. And so if you want to get an idea kind of on a general scale, what is happening to aging, the best thing you can really look at is how long that organism lives. And also to a related measurement, how healthy they are late into life. And so that's kind of a primary metric for those of us who study aging. And so that's why we'll talk about that so much. Certainly, there are many other things that are important, looking at specific organs, tissues, various molecular markers of this stuff. But this is kind of the gold standard first thing to look at if you're looking for aging. And so what Clive McKay did is he, again, fed rats with a reduced diet and found that they lived a lot longer. So that these, I don't have the numbers up here exactly, but these rats lived, I believe, about 20% longer than normal. So that's a pretty decent extension of lifespan. If the 20% in human terms would be gaining an extra 16 to 20 years of life. And if you just look at the rats, two of these rats, this is about a 3-year-old rat. So these guys are probably in their equivalent of maybe 60-year-old humans, somewhere around there. You can just look at them and tell that this guy on the right who had less food is doing a lot than the one on the left. The coat appearance is better, the posture is better. If you start to kind of look through a lot of the things that we see in human aging, generally these rats look a lot better. And then Clive McKay also had this quote, modern nutrition is governed by the philosophy that a diet which produces optimum growth in young animals is ideal. This carries with it the assumption that optimal growth means optimal health. And I think this was kind of an idea that got stuck in the mind of scientists for a long time, that that which allows you to grow faster, be stronger, is necessarily the healthiest thing you can do. And in the context of aging, that's not necessarily the case. It may be that to be the healthiest, you want to slow down some of these growth processes. We see that a lot. Things that extend your lifespan are kind of also tend to draw out development time or allow slow down that growing process during their early life. And so this idea of reducing calories was fairly new at this point. Step forward a little bit, between that time and today, dietary restriction is far and away the most successful method we've found of extending lifespan and healthy lifespan. In virtually every species we've looked at, you can do this. I have 4 species up here. These are 4 animals that are commonly used to study aging. Mice, the fruit fly, a budding yeast, which is a single cell organism, and a nematode or a roundworm called C. elegans. I study mice and C. elegans for a point of reference. And this just shows that this is a survival curve, just to orient you to this graph, because we'll see a bunch of these. So this is how many what percentage of individuals in a population is surviving at a given age. And so in this case, about half of the mice were alive around maybe 30 months of age. And if you restrict their diet, half of the mice are still alive out at maybe 45 months of age. And so you want to shift this curve to the right. That means you're living longer on average. And so you can see in all these cases that restricting diet dramatically extends lifespan. And this is just the tip of the iceberg. There's dozens of species where this has been shown. Just to be clear, I'm going to bring up the reduced diet without malnutrition. This, in various forms, and one of the complexities with this is that this could be reducing total calorie intake, some form of just like a long-term individual fast, intermittent fasting, so periodically fasting for a few hours to a few days. This, as Erin mentioned, I do a form of this. This is currently a somewhat popular diet strategy. So there look like there may be longevity benefits to this. Restricting specific components of the diet like fat, protein, or carbohydrate. Restricting even closer a single amino acid or a single molecule in the diet. So there's all sorts of things that go into this. I could go on for a long time just listing the types of dietary restrictions that are out there. So that this is a fairly broad category, but it seems to work pretty well. Throughout most of the 20th century, beyond dietary restriction, there wasn't a lot done specifically in the context of aging and longevity. Now, there was a ton of work being done on disease-specific processes, and this is kind of one area that you can highlight as public funding. So during this period, the National Institutes on Health, a whole bunch of institutes were founded that either target a specific age-associated disease like cancer, heart disease, something like that, or the National Institute on Aging in the 1970s was founded to fund research into aging itself. For better or for worse, this was also the period that gave rise to the kind of more public-facing anti-aging industry, which if you just search for anti-aging on Google or one of the other ones, this is becoming less true today, but particularly back in like the 90s and early 2000s, you get a lot of skincare products and plastic surgery and Botox and a lot of things like that. There was a lot of kind of, for lack of a better word, snake oil sorts of products out there around the anti-aging industry. And a lot of it was cosmetically focused, and many of the claims in that industry were not scientifically backed. To put it in perspective, the reason for this probably is that today this industry is kind of worth 50 to 200 billion, depending on how you measure it. And this period in the mid 20th century is when that really got going. I mentioned this for one reason, because of this industry in a lot of ways, the idea of lifespan extension kind of got a bad name for a long time up to the end of maybe the 1900s, and a lot of it was because it was kind of associated with a lot of these products that were kind of purporting making you younger when they were maybe in some cases they do give you fewer wrinkles and things like that, maybe. But so there was, I think part of this contributed to somewhat of a stagnation in aging science up until about the end of the 20th century. And that's when things really started to change. And the kind of the moment, if you ask most aging scientists that really started to change attitudes on this was this one observation in the early 90s. And up until that point, it was thought that aging was so complex that there wouldn't be necessarily a simple way to change an organism, to treat an organism and get significant change in lifespan or particular healthy lifespan. And so there's this observation that in C. elegans, this is that same roundworm that I study and that I mentioned earlier. This is what they look like. They're about 1 millimeter long. They're transparent. They have a lot of nice properties for doing aging research. They only live about 3 weeks, and they're easy to culture in the lab. So if you take this one change either the gene AGE1 or the gene DAF2, these are genes that are involved in insulin signaling, which everyone should be familiar with to some extent. You're able to extend lifespan by a pretty dramatic margin. In both of these cases, you're about doubling lifespan. So this is a major effect on aging. And so this was the observation. You can tweak one gene in an animal and get a near doubling of their lifespan. And this suggests that that lifespan in the aging process is malleable, meaning that you can do something simple to change the system and get a big benefit. And Rich Miller, who's a professor at the University of Michigan and one of the leading thinkers in the aging sciences, gave this quote that kind of puts it pretty succinctly. Until about the early 90s, it was kind of laughable that you could develop a pill that would slow aging. It was sort of science fiction trope. Recent research has shown that that pessimism is wrong. And this experiment, this set of papers, was the kind of the initial observation that started that process of changing the way we think about aging. Since that time, and into kind of the 21st century, we've, from that single gene, we've identified more than 400 genes now that you can modify to get lifespan extension, a variety of chemicals, and a bunch of processes in the kind of biological realm, which I list here. I'm not going to go through these in detail. And moving into the 21st century, there's been kind of a new revolution. The industry has grown dramatically as a science because it was realized that you really can do something about this. And Philippe Sierra at the National Institute on Aging really led the effort of this kind of new idea of geroscience, where we want to take what we know about kind of the basic biology of aging and use it to understand and treat the various diseases that are driven by aging. And so this concept of connecting the biology of aging to the processes of disease is what we call geroscience today. The other thing, this industry is rapidly growing. This is just the NIE budget since 2011 even is dramatically increased. A lot of this has come through the Alzheimer's Project Act that was started in at least 2011 and is still growing today. Goal to treat Alzheimer's disease by 2025. And the other thing that's really just happened in the last maybe 5 or 10 years is a huge explosion of interest in anti-aging medicine in the private sector. So this is just a list of a fraction of the companies that have been started just recently. So there's a huge investment in private interest in understanding these processes and developing ways to treat it. From a different perspective, we have been really successful at increasing longevity over the course of our history. If you look at kind of early human populations, we based on what we can predict, their lifespan looks something like this, where very few individuals made it past maybe their 30s or 40s. Over time, we have pushed this farther and farther till now we have an average lifespan in the United States, for example, of around 80 years. And really, if you look at the way this works, a lot of the early deaths and mortality was in this kind of early population. So one thing we've gotten really good at is dealing with infant mortality and early childhood illnesses. We've also gotten pretty good at the things that tend to cause problems in middle life. So infection, for example, and various other things. Hygiene was a big part of this. And so what we're to the point with now is most of the problems we're dealing with are these problems of late life. And we're going to talk about that more now. So why study aging today? If you look at our kind of a redraw of that curve, if we look at the structure of the population in the 1970s, around today, and in the future, what's happening is we're shifting from a pyramid shape where as we go up here, this is people who are the fraction of the population who are at different ages. And so earlier on, most of the population was young, and there was a few older people. And as we've gotten better at dealing with early life diseases, we've shifted the population more toward what we see at the end, with more and more of our population being in the older ages. And this has a consequence. If you look at most of the major causes of death in our society and look at their trajectory with age, you see that, and this is on a log scale, so this is exponential growth. All of these, the incidence and the risk of death for these diseases increases dramatically as we get older. And the consequence of that, with our population shifting toward older individuals, is that if you look at the top 10 causes of death, 6 and possibly 7 if you include influenza and other diseases. As we know in the current situation, many of these things affect elderly populations more strongly than younger populations. All of these have a shared factor in that age is the primary risk factor. It is the primary thing that tells you whether you're likely to get that disease and how well you're going to do if you do get it. And that's something like 60% of all the deaths in the United States share this property. So that's a huge factor. This has a big economic impact. Of course, we definitely care about the individual quality of life and our individual health. But if you look at how much we spend on a young person, an 18-year-old, say, it's around, a couple thousand a year. Once you get up to be 85 years or older, that spending on average goes up an order of magnitude. And so as our population becomes increasingly older, this becomes more and more of a societal, like socioeconomic burden as well. So what do we want to do? The goal of studying aging in particular is, because we see this kind of common factor, age being a primary risk factor, if we can do something to interfere with the underlying processes of aging, the goal is we can then hopefully hit many of these major diseases and improve their outcomes, improve how many people get them at the same time. And so that's the idea of the work, is to understand this underlying biology and use that to slow the aging process, and by doing that, hit all of these diseases. This again is that concept of geroscience I was talking about. Let's do a quick thought experiment just to show how effective this might be. This is a little outdated at this point, but the concept is definitely still in place. So if you take someone who is a 50-year-old woman, that's what the example is using here today. She has about 30 years of expected lifespan remaining to get out to the average of 81. If you were to completely cure cancer today, you could expect that the life expectancy of that woman to increase by about 3 years. Why is that? So just because she had cancer did not mean you affected her risk of getting heart disease or diabetes or stroke or any of the other age-associated conditions. And so just curing this one disease still leaves that person susceptible to many other conditions. Same thing with heart disease. You can do a little better curing cancer and heart disease together. Heart disease, stroke, cancer, diabetes, you get about 15 years. Slowing aging, doing something like caloric restriction, which is what this chart was based on. Potentially, based on the animal models, we could do something that would do way better than curing all of these diseases completely. And so this is kind of the motivation. We want to do something where you can increase healthy, expected, productive lifespan, and hopefully do something good to these diseases at the same time. It's always good to consider the ethical implications. This is a biology talk, but I wanted to just do a quick couple of points on this. We've talked all about the good stuff that can come out of this, but we also have to consider what the consequences are. Overpopulation is certainly a risk if everyone's living a little longer, and the downstream environmental impacts. Economic disparity, if we find an expensive drug that can extend healthy lifespan, will there be a problem where some people can't afford to get it? And I brought this up earlier, longer life without better health. One I like to address because for the most part, this is something we're concerned about. There's this concept of health span, how long you remain healthy. For the most part, things that we find extend lifespan also extend health span. And so it seems like this may not be as much of a concern as it seems on the surface, but it's something we're always monitoring. And these other ones are things that are definitely something that could be exacerbated by longer lifespan. They are also things that are already a problem that we need to deal with in the world. So there are some good to keep in mind as we kind of think through the science of making these things happen. Okay, let's go ahead and dig into the biology. What does it mean to age from a biological perspective? Well, we've actually learned a lot about this already. And it's very complicated. I mentioned earlier, basically every organ system, every cell, every molecular process gets worse as we get older. And there seem to be a set of kind of fundamental processes that at least appear to drive most of these conditions. We may add to this in the future, we don't know everything yet, but these seem to cover it. This is things like losing stem cells as we get older. Stem cells are the cells that kind of reproduce the cells in our skin and the brain and all of our other tissues. Some of you may have heard of telomeres before. We'll talk about those in a little bit. Mitochondria are the powerhouse of the cell. Primarily, they also have several other functions. So their power generation, their ability to produce cellular energy declines with age. And various other things, genome instability. So you get DNA mutations and your genome kind of becomes unstable, driving things like cancer. So we know a lot of these areas. We definitely don't have time to go through all of these. I'm going to very quickly go through two of these, telomeres and cellular senescence, just to give you a flavor of what some of the biology is underlying these things. So there's this idea in cellular aging where if you take a cell and you let it divide in cell culture outside of the body, it's also true in the body, there's a kind of a finite number of times that cell can divide. It's called the Hayflick limit. In humans, it's about 50 or 70 times. So early on in a cell's life, it's going along great, it doubles pretty regularly, but then something happens where it stops dividing. And so this is kind of always been of interest as a potential cause of aging. Maybe if our cells are just stopping to divide, we can't rejuvenate as well. And there's two concepts that I want to talk about that are related to this idea. The first is telomeres. This is probably one of the reasons our cells stop dividing, not the only one. But when our cells divide, they have to copy our DNA. And our DNA is organized into chromosomes. And at the end of each chromosome, there's this kind of protective cap, which is made up of telomeres, a kind of a repeat sequence of DNA.
George, I hate to interrupt you, but we're having some feedback and a little bit of buffering issues. So I just want to pause for just a minute.
Okay.
Okay, like you talked about, and we got a few people mentioning that too, but let's just pause for a minute and then you can resume. Thank you.
Okay. Hopefully that'll clear up soon. I haven't gotten any warnings about unstable internet connections this time. We're sounding good? Okay. All right. So we have these protective caps on our telomeres. You can kind of think of these as like the plastic cap at the end of your shoelace, for example. It helps protect the integrity of the rest of the strand. But what happens is as your cells divide, it doesn't divide it does not copy the very end of the telomere. So every time your cell divides, you lose a little bit from that telomere. So early on, it was thought that maybe this, and it probably is true to a basic extent, that this is the thing that kind of limits cell division in the dish, as we were talking about. And that might be a cause of aging. Maybe our telomeres shorten as we get older. That does happen, and that's driving aging. And so the idea behind this, that it was like the early version of this, is that telomeres are kind of a balancing act. On one hand, we need our cells to divide in order to grow and to maintain our tissues and do repair. On the other hand, if your cells divide too much, you might get cancer. So it's thought that the telomeres might be a protective mechanism against cancer, and they probably are to some extent. So to test this, we can do something to tweak this balance. In this case, start by looking at what happens if you balance toward the reducing cancer side. The way we do this is there's a enzyme in your cells that in some cells, very few, things like germ cells, like sperm or embryonic cells, some adult stem cells, cells that need to keep dividing, they have this enzyme called telomerase, which takes the DNA for the telomeres and makes them longer. So it adds back telomeres. So it undoes this shortening of telomeres with time. And so one thing you could do is to fiddle with telomerase, and then that's a way to affect telomeres. Turns out you can affect lifespan. You can completely eliminate telomeres in mice, and you'd expect that if you can't make telomerase, your cells would stop dividing sooner and sooner and sooner, and then you might get shortened lifespan. Turns out that's true. You do get shortened lifespan out at this red line, but it takes 6 generations in mice. That means you have to wait till your great-great-great-great-grand offspring before you start to see shortening lifespan. So that's not quite as dramatic as initially hoped for with this. And the other problem is that they didn't decrease the cancer burden in these cells, it actually increased it. So there's something else going on here. If you then instead push the balance the other way, so instead toward the other direction, let's say we overexpress telomerase, make longer telomeres, give all your cells the ability to make more, to keep dividing. You do get a lifespan extension. Okay, that was maybe the prediction. You get about a 20% lifespan extension. It does not make you immortal. And it doesn't affect cancer. So maybe this basic simple balance that we were thinking, just cancer versus survival, was not correct. It turns out the system is more complicated. This is the point I want to get across. All of these systems tend to end up being a lot more complex under the surface. On both sides of the balance, you have pluses and minuses. So if you have telomerase is good because it lets your cells keep dividing, but it's bad because cancers like to turn it on to become immortalized and be able to grow out of control. On the other side, telomere erosion, which can be caused by all sorts of things, means that you can't regenerate tissue as well because your cells can't divide as much. But it turns out that also increases cancer susceptibility. The reason for this is when your telomeres get too short, you kind of start to fray the end of your chromosomes, and it starts to cause genome damage, which can drive mutations, which can drive cancer. So this process was not the simple, like, silver bullet that was originally hoped to be. All right, so that's one example of how underlying biology of aging can work and how it's more complicated usually than we think on the surface. The other process I want to talk about is this process of cellular senescence, which is related to telomere maintenance. So if you have a cell in your body, it can go through a number of end phases. It can maintain its function, that's what we want. It can die. There are various ways that cells die. We lose skin cells and other cells all the time. It can start dividing out of control. This is what ends up leading to cancer. You can end up with reduced or altered function, which we get with age, or the cell can do something called senescence. And this is where it goes into kind of a non-functional state, but it stops dividing, but it's still present and alive. And so this happens a lot as we get older. If you look at Wikipedia, you see that cellular senescence is the phenomenon by which normal cells cease to divide. And yes, that's certainly true. As we'll see in a minute, it's more complicated than that, which is a theme of aging research and really all of biology. This can be caused by accumulation of cellular damage from things like radiation and oxidative species, like hydrogen peroxide buildup in the cells. On the flip side, this can be caused by an impairment of your ability to repair this sort of damage. So if cells lose their ability to defend against some of these things. And there's various other things, including telomere reduction that can cause cellular senescence. But where does it get more complicated? These cells just don't stop dividing. They start doing other things that are potentially bad. So I don't want to get into the details of this, but in senescent cells, the yellow is a bunch of things that these cells produce more of relative to non-senescent cells. So they start dumping a whole bunch of stuff out into their environment. So they go senescent and they start sending out inflammatory signals. So they're saying something's wrong, and they're also sending out growth signals. So they're not just being like good neighbors and like, okay, I'm having a problem, but I'm not going to be. They got it. No, they're screaming this around to all their neighbors and telling things are wrong and they're causing issues. And it turns out this can cause problems. So in a young tissue, all your cells are fine, they're not senescent, and things are going along smashingly. And as you get older, you start to develop senescent cells that start to dump. SASP is Senescence-associated secretory phenotype. So it dumps all these signals out, and that causes neighboring cells to start to respond to it. And sometimes it causes them to be dysfunctional. Or even worse, if you had a precancerous cell nearby, I mentioned that one of the things these senescent cells are doing is dumping growth factors out. Well, cancer cells love growth factors. They want to grow and take off, so that you can also, this can be a driver of cellular growth out of control, causing cancer. So this is bad. Senescent cells have some issues, and maybe one idea would be if we can get rid of those senescent cells, maybe we can improve the health of our other cells, our normally dividing cells and normally functioning cells, and maybe reduce cancer. And so there's been some work on this. There's initially it was done through genetic means, but now there's a class of drugs called Senolytic drugs, which are specifically designed to selectively kill senescent cells in our body, but leave our normal cells behind. If there's a bunch of mouse models that age prematurely, so they get a bunch of the signs of aging much earlier in life. These are nice because you can do experiments a lot faster. And if you clear the senescent cells in these mice, they look fairly normal. I think this is out about 12-month-old mice. Mice live about 3 years. So these are relatively young mice if they're not prematurely aged. And you can see in one of the prematurely aged mice, they have like severe kind of hunched spine. This is pretty common in mouse aging. Their fur is more matted, and they have a lot of signs of aging. This is also true in the prematurely aging mice, but even in normal mice, if you treat them with these drugs, this is an example of 2 of the drugs, cocktail that affects certain types of tissues, given every 2 weeks. This is not a continuous treatment, and that'll be important for a reason I'll mention in a moment. What you get is a pretty decent extension of the lifespan. This is maybe about 10%. just from a 2-week treatment. And I'll point out that this was started at about 750 days into the mouse's lifespan. So these mice were already middle-aged by the time they started treatment. And then it also improves various aspects of health and disease. And this is just showing 2 of these that are related to the ability of the mouse to move. So this is, I think, one of the more promising avenues that people are looking at is clearing these kind of senescent cells out as we get older and maybe being able to be more healthy. One little point, why are these even there if they're so bad for you? Why did we evolve the senescent cells? It turns out that if you look at a wound, so if you get a wound somewhere on your body, a bunch of these cells show up in that wound. So they're doing things like recruiting immune cells, which are important for healing, and they're promoting growth. So we think that senescent cells evolved as a kind of method, a tool for a body to heal. And this is just showing how big a wound is over time. And if you eliminate the senescent cells, it takes longer for those wounds to heal. So these are probably something that evolved as a healing mechanism that just accumulate erroneously with age and thus cause problems. So that's just a couple of different ways that biology is affecting us as we get older. And there are many, many, many more that we could probably do a whole series of talks just on each of those categories. So, all right, so what can we do about this? The real honest answer, of course, is we really don't know yet what we can do to extend healthy lifespan dramatically in humans. But I want to talk about some of what's there. So what about dietary restriction? We know that this works in animal models. What about humans? One of the problems with something like dietary restriction is it's not well defined and it's really complex. We don't know what kind of diet is right. There's a bunch of options there. How applicable is the animal data? So mice have a much higher metabolism than we do. They live in a different environment. They have most of this work's been done in genetically identical mice, so there's not a lot of genetic diversity. And so we don't know if it'll work for everybody. And just a major practical problem, how do we get humans to eat less on average? That's something that's a very common issue, I think. And so there is some promise for this. So there's a bunch of short-term studies that show improvements in health. There's a group of people who are doing this voluntarily, who eat less calories on their own. And there's some evidence that they look a lot younger, a lot healthier, particularly in things like heart function. And then there's a bunch of fad diets that kind of test informally some of these things. So the low carb diets and the various other reduced calorie diets. And oftentimes you see things like reduced body weight, improved insulin response, improved like blood glucose and things like that. Improved heart function is a big one. And in the animal data, that a lot of the underlying things that dietary restriction is doing seems to be the same across multiple animal species. So there's some work done. And I'm going to talk about one of these other points here in just a minute. But there's other things like you're more resilient in these animal models if you have better a reduced diet. So I make a point here that many of the diet there are drugs being designed to mimic dietary restriction, and so this might be a way around that problem of getting people to eat less if you can get a drug that does the same thing under the surface. And that brings up the question: well, how does dietary restriction work? I mean, you ask a scientist a simple question and this is what you get. So this is just in worms. There are many forms of dietary restriction. It activates all sorts of underlying pathways. It's complicated. Basically, without going through all of this, of course, there are drugs being designed to target various aspects of this, and there's lots of work being done to understand the underlying biology. Dietary restriction is affecting lots of different things, so it's not a simple one-off. So this is just an example of one thing that we're potentially you could do. Just to note some of the challenges with understanding these interventions in humans. We live a long time and we're difficult to test. It's hard to do human controlled studies. They're expensive. And because of that, most of these aging interventions have not yet been tested. They will soon, and I'm going to talk about where we are with that in just a moment. And just to be somewhat cliche, the best practices that are often recommended by your doctor are also seem to be the things that are good for aging. So if you eat a moderate healthy diet, get good exercise, these things have both been shown to extend lifespan in model systems. Again, we don't know for sure in humans. Sleep is becoming a major area. I'm surprised at just how effective getting just good sleep is. Turns out that the bacteria that grow in your mouth when you don't brush your teeth, that's been linked to age-associated disease. And maintain good mental health, be emotionally engaged, build a good social network. So it's cliche, but those are the things we know will actually help with your health and your happiness as you get older. I mentioned all of the areas of underlying aging biology. There are scientists working on all of these areas, and this is just an indication. Each of these different categories has treatments that are being studied that may affect. And what we probably want to do is combine treatments targeting a bunch of these different areas to extend lifespan. So there's lots of research going on in this area. So there's more soon. One of the things that's very promising, for the first time in history, a clinical trial has been approved by the FDA to study aging. Previously, you'd have to have if you wanted to test a drug, you'd have to be testing a specific disease, Alzheimer's, with very specific metrics. What they're doing in this one is they're taking 3,000 individuals who are at risk for multiple diseases, giving them a drug called metformin, which is very safe, it's a diabetes drug, and just watching over time what new diseases this population gets. And so the idea is if we can prevent multiple age-associated diseases that are fairly divergent in like what causes them, then that drug is probably doing something to hit that underlying biology of aging. And this just got approved, and they're doing recruitment and things like that for that. So we will be able to test drugs in humans for aging for the first time. The other thing that's new, that very state-of-the-art this year, late last year, the Dog Aging Project was started. So dogs have a particular place in our lives. We definitely want them to live longer. They are a part of our family. This is the group of scientists who's leading the effort. But the idea here is that unlike mice that we have in the lab, dogs share a lot of other parts of our environment and our diet and things like that. And they're much closer to us genetically than some of the other models we study. So if we can improve the health of a dog through a drug or something like that, we can pretty rapidly get an idea of something that might work in humans. And of course, I'm personally invested in this project. This is Doppler. He's now my 11-year-old pup, but we've had him since about 9 weeks. And so he's enrolled in this project. And if you go to dogagingproject.org, you guys can participate too. So, with that, these are just two of the many current areas of work going on in biology. And I'm going to actually just stop here and open it up for questions. This is more or less a recap of the things we've talked about today. So thank you for your attention. I'm very happy to take any of your questions.
Thanks, George. We have several questions, so I'm just going to kick those off, okay? And I'll just remind everyone that I posted in the chat area, if you want to see this recorded version on our YouTube channel, the College of Science YouTube channel, the link to the virtual Science Cafe series is there, or you can just visit science.arizona.edu. Under community, you'll find this link. But let's go ahead and I'll start with the first question from Carol. She takes a plethora of vitamins and mineral supplements, believing that should replace some of the nutrients that her body naturally loses as she ages, including a multivitamin for active seniors. Do you have any particular suggestions that might be beneficial?
Yeah, so the supplement, like dietary supplements, there is a lot of work going into those. Specific recommendations, it's hard to find exact dosing. The problem with supplements is that everyone's getting a different diet, and everyone's probably, because of our different genetics and our different background, we're all probably missing a different subset of those as we get older. There are probably some common ones. There are a bunch of these that are undergoing testing that have been tested, and there's not really one that's really nailed it as clearly extending overall healthy lifespan. Certainly for certain conditions, there will be certain specific supplements you can take. Unfortunately, I don't think I can recommend one that's going to be universally good for everyone at a particular dose. So it's really this is where personalized medicine comes in, where you really need to understand your own health state and biology to understand which specific supplements are good for you. As we start testing some of these really testing them in human aging populations, I think we'll actually be able to start answering those questions. So sorry, that's probably a disappointing answer, but that's as honest as I can be about it. So.
That's pretty reasonable. This is kind of from 3 different folks. I'm just merging it into one question. So from Sandy, Art, and Lynn, what is the best timing plan of intermittent fasting best to slow aging? And, you know, are you just reducing calories? Is that all it's based on?
So probably not. Reducing calories is, to answer the last part of that first, is probably not just reducing calories. On the intermittent fasting front, there's a lot of the biological goal is to shift your metabolism away from insulin, signal like high insulin, high blood glucose, and into more using fats as a dietary energy source in your body, which we call ketosis. And so there's a lot going on in how your body responds to extended periods without food intake. So it's probably a lot more than just reduced calories. Reduced calories is probably part of the equation, but it's probably a much more complex equation than just reduced calories. Certainly getting more carbohydrate versus protein, that seems to have a very different effect on how your body responds, how much weight you lose, what the health effects are. As to what the schedule is, again, this is something that's being studied, but we don't really know in humans. In mice, they've looked at everything from just like give them less food every day by 30 to 50% or 10 to 50%. So that's one method that works. It's probably not that severe in humans. If it works, it's probably not that severe. You can do every other day fasting in mice works. The way I do it, the kind of shortest schedule that seems to make a difference is 16-8. So you don't eat for 16 hours and then you eat for 8 hours. So you have dinner and then you don't have any food until lunch the next day. Personally, I've done everything out to a 7-day fast, and people have done the longest recorded fast in history was 482 days, which sounds unbelievable, but somebody started at like almost 500 pounds and ended at 180 pounds. And they had only water and in that case, vitamin supplements for, more than a year. What the optimum is, it's hard to say. I think this is another case where if you're interested in it and you want to try it, try a couple of different methods and see what seems to be agreeable with your biology. So, again, personal medicine, personal genetic makeup environment probably makes a big difference in what's optimal.
All right, thank you. And this is a little complex of a question. So the U of A College of Medicine Psychiatry is currently conducting studies on psilocybin, if I'm saying that right, to alleviate OCD. And can you comment on the prospects of psilocybin for research into dementia, Alzheimer's, and/or end-of-life anxiety and depression, such as those studies underway at Johns Hopkins, New York, or NYU, and Imperial College in London? Are there any proposals for such research at the U of A?
This is an area that's kind of outside of my expertise. I know a little bit about it. I know that those studies are happening. I don't know a lot of the details. The few kind of points that I've heard suggest that something like this might have some very promising applications in things like depression and post-traumatic stress disorders and things like that. In the context of aging and helping with some of the late life stuff, I really don't know what's going on. That would be interesting to find out, but I don't know the answer in detail.
Okay, thank you. Also, this is kind of combined, and you touched on it very briefly, but whenever you see aging research presented, dental health is not really presented. So you touched on it, but isn't that important, and why isn't it factored in?
Yes. So it's one of those things that just I think because aging is a relatively new and growing area, it's one of those things that people just weren't thinking about until in a large scale until somewhat recently. It's certainly important, and there's been I think it's probably among the basic set of things you should do to remain healthy as you get older. The studies that I've seen, again, this is not my particular area of focus, but the bacteria that grow in your mouth just naturally, if you let those go unchecked, some of the chemical signals they release won't just affect your gum health and things, which can have big effects on your aging, on your body, but they can also affect things like kidney function and liver function downstream. One of the big things, the areas that's important. So as we get older, our immune system and our bodies kind of improperly activate our immune system, similar to what happens with senescent cells. So you get kind of a chronic low-level inflammation. That does a couple of things. Those signals can be damaging on their own if they're not turned off properly, but they also kind of increase the threshold for like a flu infection to activate our immune system. And so doing something like letting bacteria grow unchecked in your mouth can kind of exacerbate this problem and create an even higher level of this background inflammation. So there's several different directions that can go, and that's definitely an important area.
Definitely a take-home message. Okay, so Patrick poses, if we could slow aging, wouldn't that just push the curve out further? I mean, we'd still have the heavy burden of caring for the elderly. They'd just be 120 instead of 80, plus the associated issues of having a larger population, older population, with the costs of that. So from a societal viewpoint, is the optimal solution to have everyone live perfectly healthy to the expected lifespan and then just drop dead?
Well, so that's the idea. So the idea here is sometimes called squaring the curve. So if you look at the lifespan curve, where the ideal from like not aging, like you said, would be to get out to about 50 or so, where you're still relatively healthy, and then just that's it, you're done. But of course, we're not going to do that. That comes with its own set of ethical issues. So this comes back to that idea of like, are we actually extending healthy lifespan if we extend health span? If we think about it, as we were talking, like from an economics and health expenditure point, every person in our society, we've already put a big investment on it. How many of you were productive before you were 14 years old and really contributing, giving back to society, right? So we already have a big investment into each person that's alive. And so what we want to do from a purely economic standpoint is to extend out that productive period where you're able to keep contributing, keep benefiting society, and not having a big cost. And so the idea would be to really extend out that middle period. You're still going to have problems at the end. And one answer is we don't really know how far we can push this. There's some estimates that there is some sort of maximum lifespan out there. I'm not necessarily convinced by those. It's possible that we can get good enough at this eventually to essentially reduce decline completely, and we don't know. But even if we're able to extend that, we are extending from an individual standpoint of quality of life, the number of productive, happy, healthy years we have before that kind of less fun, frail end of life period. But there's also a whole area of research that's focused on how do we compress that time. So not necessarily, when you get sick, that's the end. But if we can compress that period, extend out when you get sick up until as late as we can get it, I think there's a lot of benefit in that intervening time.
Okay, great. This is a little bit more focused. I mean, you already talked about dietary supplements. Are there any that actually improve telomere function as we age?
Yeah, so there are. One of the things that we look at is biomarkers of aging. So things that if you look at a 60-year-old or a 40-year-old, is there some metric that you can look at that will tell you something about how well that person is going to age and how long they're going to live. These things have been rather elusive, but one of the ones that people look at is telomere length. And it does, to some extent, and in some contexts, predict how long you're going to live. And so one of the things, many of the interventions that do extend lifespan have been shown to increase telomere length with age, or usually it's more of like stopping the shortening of telomeres with age, or slowing that down. Not all of the things that extend lifespan do that, so it's not universal, but that is at least a marker of an indicator of how well you're doing. Yeah.
Okay, great. So given all your knowledge on this, and I know you kind of shared a little bit about the intermittent fasting you're doing, what does your diet look like?
So I'm kind of, personally interested in diet nutrition anyway, and just kind of being as healthy and as I can be. And so my diet's changed a ton since, I probably had a typical college diet that we shouldn't talk about in any context. But definitely I've shifted over the years. I've tried a bunch of different kind of diet strategies. And what I find is that when I pick up a new diet, I will gain a couple of things that I like that make me feel better out of that diet, and I will continue with those long term. And then I'll kind of drop the big structure around it. And so I've tried probably half a dozen to a dozen different diets. But I eat a lot of vegetables, lean meat, or not lean meat, actually. So I'm kind of recently trying the ketogenic diet, for example. And so that one is more high fat, low carb, moderate protein. And there's certain aspects of that I like. And so I tend to move, the big trends are toward more natural food, less processed food, a lot more like we like to cook. So we do a lot more cooking at home, but more exercise. I think the trends all seem to be in that direction. And I definitely feel better on those diets than with a more classic American diet. Sure.
Okay, great. Patrick asked, could you talk about lifespan versus life expectancy? It just seems that the roundworms, you're increasing lifespan, but with humans, we're increasing life expectancy.
Yeah, so there's a subtle distinction here. When we're studying animals in the laboratory, we tend to have populations that were all born around the same time. So when you speak of lifespan, the whole population is kind of getting older at the same time, and you can measure that. So life expectancy that's like a population metric. How long on average does this population live? Life expectancy is a more personal metric. So if you were to ask, how long am I going to live? Based on how the population behaves, we can calculate how long you personally can expect to live based on those population numbers. And so that's a metric, and that's going to change. So when you're born, you have a certain life expectancy. Right now it's about 82 years, I think, or to 87, depending on whether you're male or female. And then as you get older, of course, that individual life expectancy, remaining life expectancy is going to go down. And when you were born, what the historical circumstances are, all that stuff's going to matter. So it's mostly that, a population level metric versus an individual metric.
Great. And then this is from Lowell. How does one volunteer for aging testing or trials?
For most of them out there, as I mentioned, have been disease specific. So if you've been diagnosed or at risk for a certain disease, like Alzheimer's disease or one of these, there are many, many trials going on that front. Right now, there is exactly one trial going on for general anti-aging. It's called TAME. I had one slide on it. TAME stands for Targeting Aging with Metformin. And you can look that up. I don't actually know the specifics of how you get involved or sign up to potentially be in that trial. It is fairly limited. I think the initial cohort is 3,000 people, and I do not know at this point if they've already selected people for that. But that's the only one out there. I think I'd probably look up their website, TAME trial, and with metformin, and there's probably information on their website about, if you're interested, how to do that.
Okay, great. And then could you discuss the value of eliminating certain foods such as sugar, gluten, animal meat? I mean, you kind of touched a little bit on that as far as trends.
Yeah, in particular, so this has changed a lot over the years. Back when I was growing up in the kind of 80s and around that time, it was all about reducing fat. It's going to give you heart disease. And so as a society, we switched over to high sugar, low fat foods. And now we have a diabetes epidemic. So there's probably some of the early ideas surrounding that have definitely changed. So fat is not considered quite so much of a boogeyman as it used to be. And right now the trend is toward low carbohydrate stuff. The answer is, again, it's pretty complex under the surface. It probably is more important what specific, like the macronutrients are. That's probably an important part of the equation, but there's lots going on. How high quality is the meat that you're getting? What is the background of the animal where that steak came from, and like the fat content? There's probably a lot of more subtle details going in that we just don't understand yet. There's been some studies in fruit flies and in mice, for example, where optimal lifespan was achieved on high carb, low protein diet, which is like the anti-Atkins, but then that doesn't seem to match with what we see in a human population where we've got a high carb diet. So yeah, we don't really understand. Nutrition is extremely complex, and because it's so public facing, there's a lot of false information or misleading information out there on it. But again, unfortunately, a lot of the answers to these questions are, we hope to know soon, but we don't know all the answers yet.
All right, and I know we're running over here, but I'll pose a couple more questions and then we'll wrap things up. I know we just continue to have, a flow of questions here, but could you talk more about the TAME study and why metformin was chosen to study that? And that question's from Doug.
Yes, excellent. So the reason that metformin was selected, we know about, there's probably a dozen or so compounds that pretty consistently, at least in the tests that have been run, extend lifespan in mice, which is usually the closest relative of humans that they've been tested in. The reason metformin was selected for this first trial on aging, one of the primary considerations is when you do a clinical trial, and especially a clinical trial where you're starting with healthy people, you don't want to cause them to get sick. And metformin has been a drug that's been used to treat diabetes. It interferes with the way our cells respond to sugar. And it's been used for probably 50 years. We have tons of safety data on it. It's very safe. It's very consistent. We know the dosing really well. That's one aspect. There's also an interesting thing. If you look at the populations of diabetic individuals on metformin, in many ways, they appear healthier than a matched group of people who are non-diabetic and not taking metformin. So something about that metformin seems to not only be dealing with their diabetes, but also giving them a bonus in a lot of other areas and making them healthier. In mice, you can, some of these areas, for example, are improved survival following cancer. Those people tend to be a little long-lived from all causes, so it seems to maybe be giving a slight lifespan extension. It's hard to say that we don't have a lot of data on that. Heart function, several other components, Alzheimer's disease. And in mice, if you give them the dose that's commonly used, maybe gives a 10% extension in overall lifespan and reduces, at least delays onsets of cancers and various other things. So it's kind of seems like a good place to start for the available drugs out there.
I have to thank you for responding so beautifully to all these questions and a fabulous presentation. We do have multiple questions that we're not going to be able to address tonight, but I can certainly direct folks to interact with you and check out your website, and then follow up with me as well. I'm on the science.arizona.edu, and I'll forward questions as I'm able. But the last question I want to ask you, I'm sure many watching are curious about you personally. What caused you to jump from an engineering career to molecular biology and aging specifically?
It really is that I got interested in aging. So when I was growing up, my dad was a bus driver, so he wasn't a scientist by any means, but he was always interested in kind of the research going on around lifespan extension. Growth hormone was a big hot topic around that time, for example. And so he would always talk about these things when I was growing up. And he always said, you should be a geneticist when you get older. That's where it's going to be. And of course, I went off and wanted to build spaceships and do research on that. But it was always kind of in the back of my mind. And then somewhere around early graduate school, I started, the interest got piqued and I started reading about it. And it really just caught my passion. And I got to the point where I was reading more about anti-aging longevity extension research than I was about my engineering work. And some light bulb clicked that maybe I should do something about it. And so I went down to the other side of campus at University of Washington and started talking to people, and ended up switching over. And that's what I do and probably will do for the rest of my career. So.
Thank you, George. We're really lucky to have you as faculty at the College of Science. And also, I know you teach our undergrads as well, the next gen you're going to inspire. And your research is exciting. And, I'm sure the community will continue to follow your work along with that of your colleagues. So thank you again for joining us. And thank you all of you that joined us virtually. We're delighted that this series was so well received. And I just posted again in the chat, if you want to forward this, or friends or family that couldn't attend, that if you'd like them to be able to see the recorded version, it's available on the College of Science website, the Science Café virtual series. We'll have links. So again, have a great evening, have a great weekend, stay cool, and thank you again, George.
Yeah, thanks for having me.
All right, with that, we're going to close out the meeting and wish everybody a good evening.